All right, we have a short one today. We're going to finish cell signaling by looking at some specific types of receptors and explore some things that can disrupt a transduction pathway. Starting with the three classes of membrane receptors. So three sort of specific kinds of receptors that you should be familiar with. And remember, what these are doing is, in general, what we already learned about. So we're just learning about a few little details about how these things can work. All right, so the three kinds we're going to look at are ligand-gated ion channels, catalytic or enzyme-linked receptors, and G-protein-linked receptors. Starting with ligand-gated ion channels. This is a mouthful, but when you look at it, it's pretty straightforward. We have a gate that is opened by the binding of our ligand, and that gate lets ions through. So remember, ions, because they're charged, cannot pass through the cell membrane on their own. They need a channel protein. And if you want to control when those ions can pass through, this is a good way to control that, right? You can have that channel, that gate closed most of the time until your chemical messenger, your ligand, binds your receptor. And in this case, this example we're going to do for muscle contraction, the ligand is acetylcholine. So acetylcholine is a neurotransmitter. It binds the receptor on this channel protein, and that will cause ultimately the channel to open. That allows sodium ions to rush into the cell, and the charge change, because sodium ions are positively charged, right? The charge differential that results from this will cause something called an action potential that will ultimately result in your muscle contracting. So this is a really ingenious way of ensuring that your muscles are going to contract when your brain wants them to, and not all the time. Because if this is not working right, and that does happen sometimes, and your muscles are contracting all the time, that obviously is not a good thing. So this is a ligand-gated ion channel. Next up is catalytic or enzyme-linked receptors. This one's kind of ingenious in my opinion. I think this is kind of a cool strategy. This is where you have sort of a double-ended protein that's kind of like doing something on either side of the membrane here, which I guess they kind of all are. But this one specifically has sort of a receptor action on the outside of the cell, and it's an enzyme with its active site on the inside. So it's kind of both, right? Okay, so we have, for example, our insulin receptor, where we're going to have our insulin binding, our ligand binding the receptor on the outside. That's going to trigger a shape change caused by the addition of these phosphate groups. So some kinase was around adding phosphate groups, right? That is going to change the shape of the active site such that the substrate on the inside of the cell, it's not insulin itself, but it's like, a response. This is response substrate, but like in a molecule that can act in response to this binding. Now it can fit in the active site, and its product will be a secondary messenger that can go on and trigger all the other things that need to happen in the cell. So it's sort of this like two-ended action here. We have our insulin binding on one side and our enzyme action on the inside, with the active site on the inside. And then here is a specific example that I see all the time. So make sure you understand this one. It's actually not different than anything else we've talked about. I mean, nothing I said for those other two really was either. But here you should know the name of it. So the receptor is linked to something called a G protein. So these proteins are sort of similar to ATP, ADP, AMP, where they're like an energy molecule, kind of. The triggering of the ligand by its binding to the receptor will cause a change in the G protein, like a change in shape. And here you can see it's like separating part of itself. And the other part of the molecule is binding to adenylyl cyclase, which you may remember is the enzyme that takes ATP to cyclic AMP, which is our favorite second messenger. So all you're really needing to know that's different from what you already learned is just that this G protein is sort of like an intermediary between, or is like specifically in this case, the intermediary between the receptor after it's bound to adenylyl cyclase. Usually it's adenylyl cyclase, which will then make cyclic AMP, which will go on to do its secondary messenger thing and cause other responses in the cells. And in this case, we have epinephrine. That's the adrenaline. We call it adrenaline colloquially, right? Epinephrine binds, and you can see on the bottom right-hand corner the constellation of responses that happen, that you're familiar with. Okay, those are the 3 types of receptors. Now let's look at what happens when things get messed up. So the disruption for this, really, I'm going to give you some specific examples of things that can happen, but anything that goes wrong in the pathway, and it's not like that hard to think of things that could go wrong, that will mess up the whole thing, right? It's it's a sort of a choreographed dance, and if any of the players in there aren't working right, it won't work. So starting with the receptor, if the receptor, there's a mutation in the gene that codes for that receptor, or it gets messed up in some way, if something else is binding the receptor, so that the ligand can't bind, then it won't be able to trigger the next step. So that's going to make it so it doesn't work, right? Here's an interesting example of this. Androgen insensitivity syndrome. This is where a fetus that has XY chromosomes, so you would typically expect the fetus to develop as male, and of course, whenever I talk about gender, I'm talking about like physiologically, right? But the organs that would normally respond to testosterone have a mutated receptor, a receptor that is not working right and won't be able to respond to testosterone. So even though they have tissue that to develop normally as testes, for example, they don't, because they aren't responding. So you'll end up with a baby that will continue down what they call the default pathway, which is female. So all babies kind of start out, or fetuses start out as female, and unless testosterone comes in to change that, you just kind of keep on going down the path. But they don't have, it's uterus and all that kind of stuff. So this is where a person will be born, and often they don't discover that anything is really wrong until puberty is not happening as you would expect it to. And then they discover oh my gosh, this is not, things didn't develop as they were sort of intended to. Okay, so that's one of them. Some cancers can be caused by receptors that should be telling cells to die off, and they don't do their job. And so that's, causing uncontrolled cell growth. And then here, just later in the process, you can have problems too. So if G proteins aren't working correctly, or any other part of a secondary messenger system isn't working, then you'll also have a problem here. So cholera does this. Cholera will permanently activate a G protein inside of your intestines. This causes too much cyclic AMP to be formed, and it will cause ion channels to be open when they're not supposed to be open. And so where you should be having sort of an osmotic situation that draws water into intestinal cells, instead, all the water is being released. And you can imagine that causes catastrophic dehydration, diarrhea and stuff, and that can actually kill people. So that is very, very sad. And just know our favorite friend, cyclic AMP, this is an important secondary messenger, and it's sort of ubiquitous, meaning it's everywhere. So a problem with cyclic AMP is going to cause a problem here. And that's it. Well done. You're finished.